Hi everyone, my name is Venise Govan. I am a research assistant here at Tennessee State University in the chemistry department, and my topic was the kinetic study of the melanogenesis from select catecholamines in the presence of cysteine or other amino acids. So melanins are dark colored pigments that can be found in all kingdoms of life. And despite many decades of intense research, there is no consensus on the precise chemical structures of these biomolecules or a precise explanation for their dark colors. There are two types of melanin. There's eumelanin and pheomelanin. Eumelanin comes from dopa, an oxidation product of tyrosine, and has a dark brown to a black color. Pheomelanin comes from a combination of dopa and cysteine and is typically reddish in color. We have reported on the effects of the presence of cysteine in the autooxidation reactions of catecholamines. In contrast to the expected reddish color, we observed that the presence of cysteine consistently results in the generation of melanin-like materials with much darker appearance compared to the autooxidation reactions in the absence of cysteine. In addition, we observed that the presence of cysteine consistently delayed the onset of color formation, and this effect appeared to be dependent on the concentration of cysteine present. In addition, there are reports of the effects of amino acids on the oxidation chemistry of catechols. Building on our earlier work, we expanded our studies on the effects of cysteine on the melanogenesis from the four catecholamines dopa, dopamine, norepinephrine, and epinephrine, which their structures can be found in the figure to the right. And we began preliminary explorations on the effects of other amino acids in these reactions. Materials and methods. So we would dissolve catecholamine in buffer, and then we would add the amino acid, which was dissolved in base, and then the reaction would happen due to air oxidation, and then we would measure this in the vis absorbance in a microplate. So this shows the results of kinetic experiments of the autooxidation of dopa in the presence of cysteine. And panels A through C show the absorbance at 280 nanometers, 315 nanometers, and 350 nanometers. And as you can see, there's definitely a delay in color formation, not necessarily a delay in the reaction, but formation of color is definitely delayed. And as shown in panel C, you can see that there's a direct correlation between the concentration of cysteine and the lag times. But if you notice in all of the panels, after the 3-hour time limit, each one of these are higher than the control group. Each one of the colors is darker than the control group. And it's notable to note, you can note that the effects of cysteine are reproducible in each one. So this one is similar for dopamine as well. There is a lag time, and it is dependent upon the concentration of cysteine present. And you can see that after the 3-hour time limit, each color is darker than the control group, once again. Now here with norepinephrine, these results, you can see that there is more variability. These are less reproducible, and you can see that each lag time is much higher. It's still dependent upon the concentration of cysteine present, but it's not the same as we saw in dopa and dopamine. You can see that at the 3-hour time limit with norepinephrine, some of them, the colors are just now starting to form, whereas before, the colors were already darker at the 3-hour time limit. And same thing here with epinephrine. Similar to the norepinephrine, the lag times are higher, the variability, and you can see that still some of the colors are starting to form at the 3-hour time limit. And you should note that dopa and dopamine were similar, whereas norepinephrine and epinephrine were similar to each other as well. In summary, for the experiments performed in triplicate, the absorbance changed in a reproducible manner as the reactions progressed. In the presence of cysteine, there was a significant delay or lag time between the start of the reaction and the observed changes in absorbance. The length of this lag time was independent of the wavelength monitored, but was dependent on the concentration of cysteine present. And the pattern of results obtained for the cases of norepinephrine or epinephrine indicate that the presence of cysteine delayed the onset of the absorbance increase at all wavelengths studied. However, the kinetic profiles obtained were much less reproducible, and the lag times between the start of the reaction and the observed increase in absorbance appeared to be significantly longer. So here, this shows the results of the kinetic experiments of the autooxidation of dopamine, but in the presence of varying amino acids. And the amino acids used were in panel A and D, serine, in panel B and E, methionine, and in panel C and F, glutamine. And here, the color formation was not delayed. The color did start forming immediately, although it was much slower than the control group. After the 3-hour time limit, they were still darker than the control group. And panels A through C just show the absorbance at 315 nanometers, and then panels D through F show the entire UV vis spectrum after the 3 hours of reaction. So in summary, when looking over the initial phases, the first hour of the various kinetic profiles shown, one can observe a pattern showing that with increasing concentrations of amino acids, the increase in absorbance is slower, but does not get delayed, as the case of cysteine. For all the amino acids tested, a broad absorbance shoulder between 360 and 380 nanometers appears in the UV vis spectra of the reaction mixtures. The intensity of this absorbance shoulder appears to increase with increasing concentration of amino acid present. The case of methionine is unique among the amino acids tested, as the UV vis spectra of the reaction mixtures containing the higher concentrations of this amino acid show a distinct absorbance band around 300 nanometers that is not present in the absence of any amino acid or in the presence of the other amino acids that were tested. In addition to the observations discussed above, the absorbance over almost the entire visible region of the electromagnetic spectrum was enhanced with increasing concentrations of the amino acids tested, particularly in the case of methionine. This gave the reaction mixtures a much darker appearance compared to the mixture without any amino acid. In conclusion, cysteine, in a concentration-dependent fashion, delays the onset of color formation during the autooxidation reaction of all 4 catecholamines tested. The presence of other amino acids slowed the reaction as judged from the disappearance of the precursor and the color formation, but did not delay it. The reaction mixtures were darker in appearance in the presence of amino acids compared to the reaction mixtures in the absence of any added amino acids. Given the importance of pigmentation in biology, any factor that may result in the change of color or a change in the intensity of color is of importance. Thank you.